Welcome to my 17th video from the 30-day challenge. In today's video, we'll be discussing the novel inventions that evolved during the viral stage of life. Hi, my name is Eduardo Hernandez. I am the creator of Emergent Chemical Evolution, the newest theory on the origin of life. I'm also the author of the book by the same name, available in all major online retailers. All right, let's get with it. Well, there are a lot of things that can be traced, that are utilized by cellular life, that can be traced through genetic investigation that originated with viruses, like the whipping tail of certain singular cellular bacterial animals that use a whipping tail to ambulate through their environment. Not ambulate, that's actually walking transport themselves through their environment. If you trace back the changes in the proteins of those mechanisms that power the the hairs that spin, you can de-evolve them into what are spiked needles utilized by viruses to invade cells. So there's another piece of evidence that shows that we evolved from viruses. Another invention from viruses that were incorporated into cellular life. One of the ones we spoke in the previous video is the thought of several virologists that consider that DNA was invented by viruses and then reintroduced into cellular life. Again, with my theory, it's obvious that we are the descendants of viruses, that viruses, once they invented the what do you call it? Once they invented the archival mechanism of DNA to protect the genetic information and make it more stable in order for the genome to grow even larger, once viruses invented that, then it allowed them to become more complex, eventually becoming cellular in nature. So that's one of the key inventions during the viral stage. Another of the inventions is the ability to have mechanisms that work at different pH levels. Again, with the system of the chaos engine transporting components through different pH levels, you're going to have the invention of components that do the same job, but do the same job in different pH levels. So you're going to have viruses that are assembled at one pH level that do the same type of work or have the same kind of biological functions that a different type of virus that assembles at another pH gradient would also have. But because of their sequence of amino acids take the conformation at that pH, then one viral type will evolve and assemble in that pH. So that's an invention of the viral stage, because that would have been hard to achieve if the genetic information and the metabolic activity would have been encapsulated in a membrane, and therefore not be readily affected by the outside environment. I believe that the cytoplasm, quote unquote, the environment, the open ocean at that time, would have allowed for the intermixing of components that reacted at different pH levels more readily than even if the membranes were primitive membranes, would allow stuff to easily come in and out. It still would have hindered the evolution of such free-flowing interchange of biological molecules at different pHs. So I think the viral stage, precellular stage, and ecosystem would have much better benefited the invention of different types of mechanisms that we still see in viruses today. We still see that they have viruses that are single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA, and that have encoding in those strands in different directions, and therefore had different types of replicating systems in place, where had cells been, or a cellular type of origin encapsulation, then shouldn't you see representatives of that type of cell still present, especially if the environment became more conducive over time for life? You would have, if encapsulation in cellular membranes was the means of the origin of life, you should still find cells with RNA and different types of replicating systems in place. You know, single-stranded cells, single-stranded RNA cells, and double-stranded RNA cells, and single-stranded DNA cells. You know, the environment changed slowly over time. There should have always been available a niche for those cells to continue to propagate and be able to exist, especially if they were able to spontaneously originate in a hostile environment. There should still be representatives of those cells today. The fact that we don't see any, and we do find abundant, much smaller and harder to find viral representatives of that type of mechanisms, screams to me that we are the progeny of viruses. And again, having a precellular ecosystem that made it readily available for viruses, which are themselves a very exponentially reproductive organism that evolves so quickly, even today, that they take seconds and minutes to replicate and evolve, is a very, very, beneficial characteristics to have that would have made it abundantly relatively easy for viruses to evolve so many different types of proteins, so many different types of mechanisms to inject their genome into other viruses, to create other types of proteins and biological mechanisms to process other proteins to create a system of energy transport, such as ATP. These things could have easily evolved in the chaos engine, passing through layers of pH with the mechanism and the currents, training those components for millions and billions of years. So the fact that we don't find in the fossil evidence cellular mechanisms until millions of years later from the formation of Earth should say something, that during that time we have black, organically laden rocks. Should tell us that we need to look smaller. We need to look at the molecular level. And maybe there's molecular fossils in those black rocks that we're just overlooking because we're looking for cellular structures. Anyway, that's my spiel on the inventions during the pre-cellular viral stage of life. So please comment, let me know what you think, and share the word. Thank you.